Welcome to your section 12.4 lecture, which will be on nervous tissue, but this time we're going to talk about glial cells. Our glial cells are also known as neuroglia. Now these are the non-excitable support cells within our central nervous system and peripheral nervous system. So the way I like to think about this is that the neuron is going to be a celebrity. So you can think of your favorite celebrity, whether that's, I don't know, Beyonce, maybe it's Kanye West, maybe it's, I don't know, Michael Bublé, whoever it is. They have a team that helps support them and give them what they need, right, in order to be successful and do their job. And so we are going to find different types of glial cells within our central nervous system and the peripheral nervous system. Just like, you know, Beyonce's housekeeper doesn't come to her for her concert, she stays within the house to do her job there and support Beyonce in that way, right? But maybe her hairdresser is going to be the one that is found at the concert. So as I go through and teach you about each of these glial cells, I'll make sure to highlight whether you'll find them in the central nervous system or the peripheral nervous system, at the concert or at the house. So before we go into that, I want to quickly mention that approximately the same number of glial cells are going to be found as there are neurons. So our glial cells will account for about half of the volume of our nervous system. But one thing I wanted to make clear too is that glial cells are going to far outnumber the neurons. So that's why I kind of like the celebrity and her entourage idea, because we have so many people within the entourage that are supporting the celebrity, and that's what's going on here. You can see even within this photo, here's our neuron, and there are several different types of support or glial cells that are assisting it. And our general characteristics for these cells is that they are capable of mitosis, they can protect and nourish the neurons, they provide physical scaffolding for the nervous tissue so that if we do have or need any help for migrating neurons that takes place during development, they can come in and guide it in that direction. And it is critical for normal function at neural synapses. Now let's talk about our first glial cell, which is the astrocyte. And astro, of course, is referring to star shape. So you can see the star shape of these cells in here, and then it is pictured in green here in this figure. And this will also be the most abundant glial cell within the central nervous system. So of course, we're going to find it in areas such as the brain and spinal cord, and they also help create the blood-brain barrier. So they have these little feet that wrap around our brain's capillaries, and it helps to control what substances can have access to our brain tissue. It also helps to regulate our tissue fluid composition, so really taking care of the chemical environment they have around the neurons, so they can regulate maybe the potassium concentration here if it needs to move ions or whatever else in order to make sure that we have the proper concentration within that tissue. And it forms a structural support for nearby neurons. It assists in neuronal development, alters synaptic activity, so if we need to add, eliminate, or influence any of that synaptic activity, and occupy space for dying neurons. So it does quite a bit for our body, which is why it is so abundant. Now let's take a look at our other cells that we find within the central nervous system. So this is going to make 4 total glial cells for our central nervous systems. Those astrocytes we just talked about. Then we'll talk about our epidemial cells, microglia, and oligodendrocytes. So our epidemial cells, you could see pictured over here, are going to help to line cavities within the brain and spinal cord. And when I talk about cavities, I am talking about ventricles, which we will discuss more within chapter 13. They are going to serve as a part of the choroid plexus that we find within the ventricle, which is responsible for producing cerebrospinal fluid. And then we have our microglial cells. So you see them in orange over here. I like to call these our nursing cells because their job, even though they're small, they wander around the central nervous system and they are going to replicate in infection. And they are basically your brain's macrophages. They are going to be phagocytic cells, so they eat other bad cells, and they engulf those infectious agents and help to remove any debris. And then lastly, we have your oligodendrocytes. You could see it pictured over here. I know it kind of blends, but these oligodendrocytes have these slender extensions that will wrap themselves around the axons of our neurons in order to form myelin sheaths. So you can see this illustrated down over here, our oligodendrocytes creating those myelin sheaths for the axons of the neuron over here. Now let's talk about our glial cells within the peripheral nervous system. We only have two glial cells here, which are our satellite cells and the Lemocytes, which are also known as our Schwann cells. So our satellite cells, we're still learning more about them, but they are going to arrange themselves around our neuronal cell body. So here we see a unipolar neuron, which is going to be our sensory neuron here, and the cell body is found within the ganglion. And they are going to help to electrically insulate and regulate the exchange of nutrients and waste by doing this. Next type of cell are our Lemocytes, or our Schwann cells. I tend to call these Schwann cells just because that's what they're mostly referred to, but you should know both names. And these are going to be elongated flat cells that will ensheath our peripheral nervous system axons with myelin. We will only have one of these cells wrap around and make one myelin sheath. And this always reminds me of when I was a kid. I used to take a fruit roll-up and kind of wrap it around my thumb, and I would eat it in that way. So it just keeps going until it kind of finishes up. And then you'll see that that nucleus will shift to one side. And by having this myelin sheath in place, it's going to allow for faster action potential to move through. That is because it insulates the axon. And now for a clinical view on tumors of our central nervous system. When we do have a tumor, we can call this a neoplasm, and this is going to take place from unregulated cell growth. And sometimes it occurs in our central nervous system, and other times it can happen outside of that. But our tumors that are originating from the brain or primary brain tumors are what we call neoplasms. And typically they're going to originate from supporting tissues, so tissues that have the capacity to undergo mitosis. So that wouldn't be the neurons themselves, but can be from the meninges, which are our protective membranes of our central nervous system. And we'll discuss those later on. And they can also be from our glial cells, which are going to be the neuroglia, those supporting cells. If that tumor does develop from glial cells, then we call this a glioma. This may be benign, but it can also be malignant, meaning that it's capable of metastasizing and spreading to other parts of the body. Next, we're going to talk about myelination. So that is the process of wrapping an axon with myelin. So what is myelin? It is going to be several layers, which we see over here, of membrane of our glial cells. And it has a high lipid content that gives it a glossy white appearance and insulates our axons. The glial cells that help us do this in the peripheral nervous system are those Lemocytes, also known as Schwann cells, and that's what you see pictured over here. They're wrapping themselves around that axon. But in the central nervous system, we have a different cell that does this, and that's the oligodendrocyte. So first, let's talk about those Lemocytes in the PNS. It's going to encircle the neuron's axon and wrap it in layers, forming that myelin sheath. The Lemocytes cytoplasm and the nucleus are going to be pushed to the periphery. So as you're watching these multiple photos of this process taking place, you can always see that that nucleus and the cytoplasm is shifted to one side around the periphery of that myelin sheath. And this forms a neurylema. Remember, anything that has that lemma ending refers to like a plasma membrane. So this is the plasma membrane of the Schwann cell, or the myelin, really, that's wrapping the axon. The neurolemocyte can myelinate just 1 millimeter of axon, so we're going to have several of these neurolemocytes that are needed for 1 axon. And you'll notice in the next photo that we are going to have some gaps that form in between these myelin sheaths, and these spaces or gaps are known as neurofibrillal nodes, or nodes of Ranvier. And here's that photo here. So you can see these spaces or gaps in here are nodes of Ranvier, or we can also call them the neurofibrillal node. And here are the neurolemocytes or Schwann cells that are making up the myelin sheaths, and we see that nucleus shifted to one side. Now let's talk about myelination by our oligodendrocytes within our central nervous system. So we're going to see that one oligodendrocyte is myelinating 1 millimeter of multiple axons at each of the multiple spots. So we see that it has these processes that extend on out and can myelinate several axons at one time. In this way, we have no neurolemma that's formed, and neurofibrillal nodes between adjacent wrapped segments are existing once again. So here's one here, there's another one there, and another, several of them. For unmyelinated axons within our peripheral nervous system, our axon is going to sit kind of depressed within a portion of a neurolemocyte. So we could see this taking place over here, all of those axons grouped together, or you can see them kind of enmeshed within the neurolemocytes in several spots around the periphery here. Within our central nervous system, our unmyelinated axons are not going to be associated with oligodendrocytes, so they are just one continuous axon. Now for another clinical view, we'll talk about some nervous system disorders affecting myelin. First, we have multiple sclerosis, and this is going to be progressive demyelination of neurons within our central nervous system. This is an autoimmune disorder where the oligodendrocytes are attacked by our immune cells, and we have repeated inflammatory events causing scarring and permanent loss of function. You could see this displayed on this transverse cross-section of the brain, where we see these plaques that have developed in here. Now we can have a similar thing happen within our peripheral nervous system in the Guillain-Barre Syndrome, or GBS. This is when we have a loss of melanin within the peripheral nerves due to inflammation, and that leads to muscle weakness that begins in distal limbs and advances to our proximal muscles. And most function is going to be recovered with little medical intervention.